Morning, everyone. Welcome. I'm Cassandra Albright from the University of Pittsburgh. And on behalf of the RCMB Web Committee, thanks so much for being here. We're really excited to have you all here and to welcome two really great speakers for our RCMB Toolkit series. Today, we'll be discussing the Human Lung Cell Atlas and LungMAP, two open access tools with a wealth of opportunity for data discovery. We'll have a talk discussing each in greater detail with some time for discussion and Q&A at the end of the seminar. And then in addition, our speakers are happy to take questions during their talks to have an interactive discussion as much as possible. We're really excited because there's so many of you here, but given that, please feel free to type any questions in the chat box or to raise your hand so that we can unmute you to be able to ask questions as we go along. And then with that, let's get right into our talks. We have plenty of time. I'm going to go ahead and introduce our first speaker. So Dr. Lukin is a group leader at the Institutes for Lung Health and Immunity and the Institute of Computational Biology at Helmholtz Munich. There he leads the Integrative Genomics Lab, which uses machine learning and single-cell technologies to build population-level reference atlases and derive patient-level insights from these atlases. The Lukin Lab applies these approaches to the healthy lung and various diseases, including COVID-19, COPD, and ILD. He's also the co-founder of the Living Benchmarking Project, Open Problems and Single-Cell Analysis, which aims to formalize tasks in single-cell data science and set quality standards for the evaluation of these tasks. And then at the Human Cell Atlas, he leads the integration team, focusing on building integrated single-cell reference atlases for the HCA bionetwork organs and tissues. Please feel free to get started, and thank you very much, Dr. Lukin.
Thank you very much for the kind introduction and the opportunity to give a talk here today. Hi, my name is Malta, and I'm as mentioned, kind of at Helmholtz Munich, between computational biology and pulmonary research. And in the past approximately three and a half years, we've been working on building a reference atlas of the human lung, trying to combine the expertise that we had locally on computational biology, as well as leveraging as much of the community expertise as we could to really reach out beyond our borders and getting a lot of very positive feedback and help also with building this community resource. But before I dive into the Human Lung Cell Atlas, I quickly want to introduce the lab that I lead. We work on translating machine learning models on single cell data to clinical applications in lung research. And there are three pillars within the lab, which start with, on the one hand, we work on benchmarking and pipelines to develop kind of quality standards in single cell analysis. And with that, within the kind of framework of doing this, we're trying to get as many machine learning scientists as possible into single cell and to tackle relevant biomedical problems, even if they don't have the background, to really make sure we can leverage this expertise. As a second pillar is we're building reference atlases, as I'm going to talk to you about a little bit today as well. And we try to, for the lung, but also for other tissues and organs. And then as a third pillar, we are really trying to leverage these atlases to work on clinical data sets to understand biological processes as well as patient variation. So the lung is a comparatively well-studied organ on the single cell level, given its accessibility. There are many different data sets out there. So if I want to understand where my favorite gene is expressed, I could go to any one of these. However, they will disagree. And so what we've been working on previously is try to leverage these individual data sets and bring them together to build a consensus view of what the healthy human lung looks like. Asking multiple data sets at the same time, particular questions, has many different advantages. So this is something we call aggregated queries. On the one hand, what we've done previously, we can look across these data sets to robustly answer the question, which cell types express entry factors, for example, for SARS-CoV-2. We can ask whether a cellular state that we find in one study is specific to this disease that is being studied or this clinical phenotype, or if it actually can be found across diseases, maybe across data sets, across populations. And we can study inter-individual variation. And this is particularly relevant to do across data sets because up till 2022, the median number of donors in a single cell data set was 11. That's really not a lot to be able to understand how individuals differ in their cellular profiles. And we need to do this at a scale that is only achievable by combining these data sets. And then there's been a really nice paper a couple years ago now, it's 2024 already, which had a more visionary outlook on how these reference atlases, these aggregated queries across data sets, can impact biomedical applications, going from predicting pathogen entry factors to modeling disease prognosis and progression. The challenge with doing this per data set across many data sets without combining them in some way is that the labels, my cell type labels will disagree between data sets. What I call a goblet cell, you may call a club cell. And this is true, we found actually across the board that this agreement on where this continuum kind of changes differs between data sets. And this is just one example. Also, there are batch effects between studies. Batch effects are technical artifacts in the data that arise only because of the experimental design choices that were made to design to generate this data set. To give you an illustration of what that is, if I have a data set where I'm kind of collecting samples from patient one that comes into clinic one at time point one, and I dissociate that tissue, I collect the cells, I measure the transcriptomes, and in the process of analyzing this data, I map this into a low-dimensional space, and I see this looks great. My cells separate based on their identity, and I can analyze that. However, typically we recruit, potentially at multiple sites, multiple individuals, different times. And so if a second individual is recruited to the study and we do the same process, we map this to the same low-dimensional space, we will find that these cells now differ not only based on their identity, but also based on what we call a batch covariate. And this batch can be related to technical effects. There might be different levels of ambient RNA. The way the samples were processed might be different or handled as well. But this could also be differences in biological biological phenotypes of these samples that we may want to not preserve to be able to analyze this jointly and perform one of these aggregated queries. These samples may be taken from slightly different locations in the tissue. Might be different individuals, different genotypes, of course they differ. But in order to be able to analyze this together, we require that these batch effects be removed and the data set overlaps so that we can really perform a joint query of that data in the end across samples, across individuals, across data sets. Fortunately, this is possible with different methods. There are in fact over 200 different data integration methods that have been published in the single cell field. And this is just a kind of an overview over some of the more popular ones. At least they were very popular approximately 2 to 3 years ago when we started this study. And what they of course give you different results, right? So none of these methods will do exactly the same thing. They've all been published. So just to give an example of how that looks like, if I take one of the first atlases of the human lung, this is a data set that was generated across 3 different sites, 3 different data sets. And I just take this data set and I color the cells on this UMAP by cell type. And if I just focus on one of those cell types here that have been annotated, macrophages in this case, this light blue cluster, I find this in 4 different locations on this UMAP. If I look more deeply into this, these are from different donors and from different data sets. This is a batch effect that precludes me from analyzing this data set jointly across the population. Now, if I apply 3 of these different methods, I'll get 3 different results. And this is somewhat frustrating when we're trying to choose a method to integrate these data sets. If I focus again on macrophages, in some cases I get something that I do not expect at all. Is probably a bit strange to interpret. These kind of longer, elongated clusters in 2 of them. In other cases, I get a distinct cluster. In other cases, this cluster may overlap with other cells that have been annotated. Now, I cannot go through 200 versions of this for every data set that I'm integrating. So what we really require is metrics that measure successful integration performance. This is what we initially set out to do. We set out to understand how to best integrate multiple data sets, multiple single cell data sets of human lung samples. But we started to kind of generalize this problem a little bit more, leveraging published single cell studies to curate 86 different batches of data in 13 different tasks. We processed these data sets in a consistent way, and then used 16 of the more popular integration methods at the time. That gave us these 3 different outputs, as well as over 500 different ones. And then we developed metrics, 14 different metrics that measured how well batches integrate, but also how well biological signals that we care about are retained. such as whether cell types can be separated, whether trajectories are still defined after integration as well as before. And that gave us kind of an overview, like these long tables of how different methods perform at the integration. Now, we have a lot of data sets. We have a way of assessing how to best integrate them. What remained for us, and this is something that Lisa Sikkim, a very talented PhD student who I've had the fortune of working with, was pushing, is putting these two things together to build an integrated lung cell atlas. We started by selecting 14 data sets, which we regarded as high quality, which were landmarks data sets in the field at the time, pre-processed these consistently, harmonized the annotations of these cells to make sure that we could at least evaluate how different methods compare to one another. We also ensured to harmonize metadata. And you can imagine this is not a straightforward process if some people record smoking status as has never smoked or has ever smoked, and others record former or current smokers. These two things aren't really as compatible. So we relied a lot on people providing metadata in a way that we could use across all of these studies, and brought that together in a way to have like a metadata on 166 samples in the core. And then we also worked to annotate exactly where these samples had been taken from, using just a simple coordinate framework, with 0 being samples from the nose and 1 being parenchymal samples, with the scale between that depending on the generations in the trachea, for example. Combining that with our favorite integration method, based on based on our performance ranking, we generated a core of the human lung cell atlas, which is initially just a reference embedding of 14 different single cell studies. At first glance, this looked quite nice. So we could separate, major cell populations, and that was great. But we quickly noticed that we are definitely not the experts in lung biology in all of these cell lineages, especially. So we reached out to the Human Cell Atlas Lung Bio Network, specifically the people you see here at the bottom, who helped to annotate all of these cells and gave us kind of a consensus of 61 cell identities based on input from all of these 6 annotators, effectively, who were very instrumental to the study. And that we ensured that each annotated cell type or cell identity came from multiple individuals, multiple data sets, so that we were sure that this is not an artifact of a particular data set, particular sample, an outlier, effectively. Now we have reference embedding of human lung cells, which are annotated to 61 kind of consensus cell identities. That's great. What can we do with that? And that's really what I want to talk to you a little bit more about. On the one hand, we could recover cell types that were not recoverable in any individual study. Specifically, we've recovered 9 rare cell types, a few of them had not been described in the adult human lung. And this is because we suddenly didn't have 50,000 cells, we had nearly 600,000 cells from over 100 individuals. And for example, you can see here, migratory dendritic cells were very clearly identifiable based on marker gene expression, but were actually not annotated in these original studies. We could associate genome-wide loci from genome-wide association studies to particular cell types based on the proximity of these loci to marker genes of these cell types to provide mechanistic hypotheses. And this is something which, of course, needs to be validated, but we found that without using our reannotation, with no signal could be recovered here. So we can't just do that over data sets. We need to make sure that this is a batch corrected and reannotated space. And then two aspects I want to tell you a little bit more about. We could model all of these metadata that we'd worked hard to harmonize. We could model the effects of those metadata on cellular transcriptomes. And so this is what we call gene module detection. What this boils down to is, for example, this kind of common coordinate framework axis that we assigned to the samples, being 0 being brain proximal samples from the nose, versus 1 being very distal samples. We could now assess how this, by modeling the gene expression per cell type, modeling how this affected the transcriptome of the same cell type. And we found consistent signals across airway epithelial cells, which were enriched for particular terms. Now, I don't want you to have to go through all of these individual enrichment terms here, but to summarize, we find that towards the proximal axis, we find increased antigen representation across airway epithelial cells, increased keratinization cornification processes, and oxidative force correlation, all of which might be related to the more higher exposure to the outer environment, and therefore upregulation of these processes as a consequence. Towards the more distal axis, we find that specifically in multiciliated cells, lung developmental pathways were upregulated compared to the same cells closer to the nasal cavity. And so this is something we could do across all of the different covariates which we collected, which was quite nice, that we could find signatures effectively that identified anatomical, like this kind of axis, as well as signatures for BMI, as in specifically plasma cells, which we reported, and several other covariates that are associated with demographics. But maybe the most exciting and the use of the human lung atlas that I'm going to focus on for the rest of the time is that in the same way that the Human Genome Project delivered a genome reference, which enables us to rapidly align new sequencing reads against, and therefore, no longer require putting together a new genome every time we want to analyze sequencing data. We find that the Human Lung Cell Atlas, or reference atlases like the Human Lung Cell Atlas as well, enable a rapid analysis of new data sets by projecting that data onto the reference atlas and transferring information from the reference atlas, this kind of very richly annotated data set, to the new data sets. The way that works is through a method which we published a couple of years ago called single-cell architectural surgery. And I don't want to go too much into the detail of this method, but conceptually what happens if we have the Human Lung Cell Atlas as one data set over here, and we have a new data set we want to map this onto, we use the same model that we trained all the data set we trained on the Human Lung Cell Atlas to integrate all of these data sets, and only adapt it slightly for it to be able to take a new batch of data in, which leads to the new data set mapping on top of the Human Lung Cell Atlas into the same informative space. If you're interested, please take a look at the original method that I'm just going to highlighting here briefly, which was published a couple of years ago. What that does effectively, so if we test this on a new data set, and this is a really nice study published by the Teichmann and Meyer labs in Nature Genetics a couple of years ago, where they found used spatial information as well as single cell data to annotate 77 cell types in the human lung across multiple locations. When we map this data set, which was not available at the time when we kind of generated, when we put together the Human Lung Cell Atlas, onto the Human Lung Cell Atlas, we find in here, you see the cells from this new data set in black and everything else colored based on the annotations we have at a particular resolution. So we have 5 different resolutions of annotations. We find that this maps fairly well, just at visual inspection at least, to the Human Lung Cell Atlas. And this mapping alone automates several pre-processing steps that would otherwise need to be done, such as feature selection, dimensionality reduction, and helps us already partly along the way of analyzing this new data set. The power of this mapping is really, though, in transferring information from the reference atlas, the Human Lung Cell Atlas, to the new data set. And this works by very simple concepts of similarity and majority voting. If I kind of schematically assume that these are cells that I have in the Human Lung Cell Atlas, and I'm just going to call them generically cell type A and cell type B, I can map to this space a new cell, assess what are the closest similar cells, and then by majority voting, I can say this is probably a cell of cell type A, given that it maps into this space. Similarly, I can say this is a cell of cell type B if it maps into the outside here. When it becomes really interesting is when we are uncertain. If a new cell is mapped into this space and we are not sure whether this cell should be assigned cell of cell type A or B, there can be many different reasons, several different reasons. On the one hand, this could be a residual batch effect. We have not properly corrected for the technical differences, and that is possible. If that is the case, then every cell from this new data set will have this uncertainty, and we will not have done a good job, and we'll find that very clearly by looking across all the cells that we mapped to the human lung cell atlas. This can be a cell that should actually be exactly here, but is transitioning potentially between these two cell types. So this uncertainty suddenly becomes informative if we are uncertain about what label to give it. This could be a new cell identity that we haven't seen before, either a new type or a cell that is expressing cellular programs that were are not present in the human lung cell atlas in the kind of this healthy reference. All of these last three cases are informative, and we can easily rule out the first by looking at all cells from a new data set. So when we apply this same analysis of label transfer and uncertainty quantification to the data set I just introduced, we find, and then this is colored by kind of the darker, the more confident we are about the labels that would be transferred. You can see that in some cases, so overall, first we know the labels of this. We transfer, approximately 65% of labels correctly based on the annotations of that data set, which we know. And we are uncertain in approximately 20% of cases. This is particular here, where you can see that these cells have a high uncertainty, and that's because they're not actually overlapping with anything in the human lung cell atlas. These happen to be erythrocytes, which we removed from the HLCA because they were not of interest to the study at the time. If we break this down per cell type, we find that the performance of the label transfer is actually very dependent on that cell type. For example, this kind of club, goblet, or kind of other secretory cell state continuum does not seem to agree between the reference atlas, the human lung cell atlas, and the new data set. Now, this might just be that it's very difficult to really define these boundaries in what is otherwise maybe a continuum cell state. But what is particularly noteworthy here is that most of the cases where we actually perform very poorly is where we expect to perform poorly. These cell types or cell identities at the end, they are not present in the human lung cell atlas. So of course, we are very uncertain about what, and we don't do a correct job of mapping this new cell type because the label, the cell type, because we do not have that cell type annotation in the reference atlas. In fact, this performance of approximately 65%, when we look across studies that we integrated, was very competitive with how individual studies had annotated their data sets compared to the consensus reannotation. So we found that approximately only 40% of cells were initially annotated in exactly the same way that we annotated them based on this consensus from 6 different labs. And even though it's competitive, it's much, much faster. So what I'm doing here takes me a few hours rather than annotating every cell, pre-processing a new data set, which can take days, weeks, months, you name it, depending on the experience of the person doing this job. So we can do this with one data set. We thought we might as well do it with many. So we extended the human lung cell atlas with over, I think, 40 data sets, 46 data sets from the original 14. And we wanted to understand, does this work all the time, or is there particular cases where this label transfer just doesn't work? And we looked at this by assessing the uncertainty in the label transfer across all cells and looked at different covariates that might explain higher or lower uncertainty in this process. And the only covariate we found, so we could calibrate that, good uncertain, like a good label transfer performance was an uncertainty under 0.2. The only covariate we found that explained higher uncertainty in particular data sets was disease, and specifically the COVID data sets, which we had mapped to the healthy lung cell atlas, showed much higher uncertainty. While on the other hand, this worked very well for single nucleus data, other single cell data sets, various sampling methods, different technologies, and most diseases, except for these COVID-19 data sets. Now, what is important to consider here is that the early single cell data sets of COVID-19 were very severe very severe cases. So actually, when we looked at how that breaks down, this uncertainty per cell type, we find that the cell types where we are very uncertain of the label we should assign them when mapping new data to the reference atlas were predominantly myeloid cells or macrophages, non-monocyte derived macrophages and monocytes. And these are particularly affected in severe COVID and notably have a different phenotype than macrophages found in the healthy lung. So with that, we kind of concluded for us that the human lung cell atlas also functions as a transcriptomic reference for COVID-19 cases, specifically because most of these cell types actually do map very well. And also, uncertainty will be able to highlight disease-associated cell states. Now, this is a first hint we got that maybe this label transfer uncertainty highlights where it's interesting to focus on. So which cell states are particularly disease-associated or affected by disease. And so we tested this on an IPF data set from the Shepherd lab, which was published, I think, in 2019 or 2020, where we mapped this data set, including we had the annotations. We knew what should be the annotation that was mapped. And we just tested how does the uncertainty of the label which we would give that new cell, how that looks between cells from healthy or from normal tissue versus cells from disease samples, so from patient samples. And at first we observed, kind of looking at this plot of the uncertainty in this label transfer across cell types, is that the cell types that had the largest disparity between in uncertainty between the cases and the controls were cell types that are associated specifically with IPF. So where there's been several studies to show that alveolar fibroblasts and macrophages express different programs in IPF than they do in normal individuals. And in fact, looking more detailed at this, we found that these are actually different cell states, which we capture based on this continuum. However, there's still high uncertainty cells in IPF patients, as well as low uncertainty cells. So what is this effect of uncertainty? Is this just us being unsure about what label this is? Or are these actually different subtypes of cells? And when comparing these high uncertainty to low uncertainty cells, we find that this score actually kind of extends to separate healthy cells from normal tissue, which we call healthy here, to kind of give you a gradient. And that shows us a disease signature score, that the higher uncertainty cells or the cells which we are uncertain of the label which we transfer in IPF patient and IPF samples highlight a disease-associated cell state, and compared to the low uncertainty cells, which highlight actually a more homeostatic cellular phenotype. So this enables us to kind of use label propagation to immediately highlight in new data sets. Remember, we're doing this by mapping a new data set to the human lung cell atlas, immediately transferring this information from the lung cell atlas. And then that highlights, with this uncertainty analysis, which states of cells are particularly interesting to look at, to follow up on. So very limited manual data processing is needed at this point. And we've gone effectively more than 50–60% through a data analysis pipeline in the matter of a computational kind of time frame of just a few hours using one method. So with that, I want to tell you a little bit more about how you can use the human lung cell atlas to either explore it or use it. So if you look at this QR code, this will take you to the Cell by Gene platform, where you can start one of these browsers here to immediately explore which genes are expressed in which cell types, for example, across the human lung cell atlas, or just take a look a little bit more at the annotations of the different resolutions. If you go to that link, what you kind of are first confronted with is a browser like this. If you just click explore, I believe, or rather than download, these are the two options that gives you. On the left-hand side of this browser, in case you're not familiar with Cell by Gene, this is something developed by the Trans-Zuckerberg Initiative. On the left, you have all the different covariates which you can color this cellular representation by. For example, here I'm coloring by the third level of annotations we have, and that shows you kind of a separation of cells according to cell type. On the right-hand side, you can input the genes you care to check, where they're expressed. So, for example, if you have a new study, you're confused by this particular cell type or cluster that you have in your data, and you know the marker that's in there, you can input that marker in there and see where it falls within the human lung cell atlas to see what label might be transferable there. For example, here I'm just kind of inputting FOXI1, and immediately I see here it highlights a particular area of this UMAP representation. But because we don't want to rely on looking at UMAPs only, we can look at across this annotation levels to find that the distribution of FOXI1 expression seems to be within pulmonary ionocytes. And that immediately highlights, okay, my favorite gene I've looked at here is specifically expressed in ionocytes, and that gives me a hint of where I can look further. And of course, there are 5 different resolutions of hierarchy, so you can always go deeper or more coarse depending on where you want to explore. So that is kind of the how to explore the human lung cell atlas in terms of asking which gene is expressed in which cell type. You can also do limited differential expression testing there, looking at which genes are specifically differentially expressed between 2 cell types, and just kind of exploring the space, highlighting particular areas, subsetting, etc. But I've talked to you a lot about reference mapping, so how to compare or map your own data set to the human lung cell atlas and transfer information. And that's all great if you need, someone who knows exactly what they're doing computationally to do that. And so this is why we have placed the human lung cell atlas on initially 2 different or 3 different platforms. And with this, I was kind of want to announce also the beta release of a new platform, which does not require you to do any programmatic access to the human lung cell atlas, and basically uses in the browser, allows you to map your own data sets to the human lung cell atlas to do basically everything I just told you about in terms of this uncertainty quantification. Of course, the human lung cell atlas is available on Azimuth, which is probably the most widely used platform for this purpose. We also uploaded it to Fast Genomics, which is a company at the time we were working with. I think maybe the free usage of that platform has maybe expired by now, and this is why today we're kind of pre-releasing archmap.bio, which you can kind of access immediately by going to this, looking at this QR code over here. Archmap.bio is a project which I contributed to, which really kind of was started by Molot Falahi, a former PhD student in my former lab, and now who's now based at Sanger. And this is effectively a browser-based platform to perform this reference mapping for the human lung cell to the human lung cell atlas, as well as many different cell atlases. The way this works is that optionally you can sign up. You don't have to, but there are some benefits to signing up because then you get your own space on archmap.bio. You then start to kind of start a mapping process where you can pick an atlas which you want to map to. At the moment, there are 10 core atlases. This is still, as a beta release, is still in development. So we're working to add more. We're looking to get more descriptive, give you more information on the ones that we have in there already. You can pick a model that was used to train this, and then pick a classifier. You don't have to pick the classifier. There are defaults set for all of these. And then you just upload your data. Here you can kind of see what choice was made here. Here, for example, someone is trying to map to the retinal atlas using the SCVI model and an XGBoost classifier, where this is completely irrelevant if you don't want to pick these things, as long as you know you want to map to the retina. And then you can just drag your file here where it tells you exactly how that file needs to be formatted to be able to perform this mapping. And then you start, and it kind of gives you a bar here with the progress of this mapping process. After mapping, you get a representation in the same way that I just showed you on cell by gene, with a kind of a browser-based cell by gene instance, where you can see how your data set maps to the atlas you chose to map it to. And it automatically also has this uncertainty quantification. So you can look in the same way that I just showed you where FOXI1 was expressed, you can look which cells are particularly uncertain in the labels, which cells that were mapped, where they fall on this in this UMAP space, but also which cells you may want to take a closer look at in this. And of course, if you do decide to sign up, you can do this in a kind of a community. You can set up your own community in there where you can share these results and work on them with your team to add annotations to your data set in the browser. And here, for example, there's an institution. You can just sign that institution up. If you're the person who starts your institution on there, you're the admin of that institution, and build teams within that institution. And within these teams and institutions, you can share privately these results, or you can choose to make it more share across teams, etc. You can even upload your own atlas, which you may want to map to, and you can collaborate with other between teams or between members of your team on this cell hygiene browser. And to show you that it is actually fairly straightforward, here's a quick video for just logging in and going through these steps that I just showed you. So here, Chelsea, who's now currently leading this project, is quickly logging in, waiting to load. Obviously, she's done some mappings before, and she can add a new mapping. In this new mapping, she then kind of goes through the different atlases, which she would like to use, but there is basic information on most of these atlases, at least that are currently in use. And she's selecting human lung cell atlas, picking a model in the classifier, just the first thing that comes up, and then clicking on selecting her data sets for upload, going to a file which she already has prepared, specifically using the Delory et al. Postmortem COVID dataset, and then going to this. Maybe that was a little bit fast, but effectively going to this, area where it immediately shows you the bar of progress. And because that takes some time, sometimes, depending on the server load, it can be very quick, and depending on the size of your data sets as well, between 10 minutes and a couple of hours, or maybe, half a day if if there's a lot of people on that server. And then you can kind of look at this afterwards, once it's mapped in this space. And here again, kind of color by different information, which you have in the atlas. And here, for example, she's highlighting that the query data set, all the cells that are in the query data sets, the new data set that she's mapped to the reference atlas, and highlighting immediately the uncertainty in the label projection. So this, for example, highlights that I believe endothelial cells are down here. There are a lot of cells from this new data set, where we are very uncertain, or where the reference mapping process is uncertain what cell type label this should be given. And that highlights that there might be a distinction between the endothelial cells of this post-mortem COVID dataset to the healthy human lung cell atlas reference, to kind of highlight where it might be worth taking a closer look. And with that, I want to summarize. We've built the human lung cell atlas as kind of an integrated reference overcoming batch effects between datasets, leveraging this community input for annotation, and also very happy to receive further community input if you think our annotations are rubbish. This is something we're working on actively to improve. The reference atlas offers a lot of unique opportunities to kind of use the scale in terms of the cells, but also the number of individuals represented to model inter-individual variation, to capture rare cell types, but also to analyze new data sets and then compare different data sets based on this mapping. RCMAP is a new browser-based tool where you can access the human lung cell atlas reference mapping without the need for programmatic or any computational expertise. And just as a quick outlook, I like giving outlooks at the end of my talk. We are currently working on the second version of this, focusing on healthy and COPD samples. If you're interested in contributing, we're very happy to. Please, contact me. I'm happy to see anybody interested. And we can use as much community input as possible. And with that, I want to acknowledge a lot of people who did a lot of work on this. First and foremost, kind of Lisa, who is the driving force behind the human lung cell atlas by pushing this project. This happened in Fabian's lab, who really kind of gave us the framework for being able to do this. And without Sasha Masharin and Martijn Navijn, this would have been a very sad computational effort, and not at all grounded in the biology and the lung biology that they made sure that this really was, and really co-led this study with a lot of very, very useful input. And of course, here I'm highlighting in bold predominantly the people who contributed unpublished data sets, which really made sure that this human lung cell atlas is relevant also, for studies that have been published since, and can be used in this way. And the team, the RCMAP team, we've been working on this quite a lot in the past 2 years approximately to make this platform a reality. Thank you very much. I'm happy to answer any questions.
Great. Thank you so much for this talk, Dr. Lucan. It's not often that we get like a beta release on our webinars. I'm pretty excited too, as soon as this is over, run off and play with it myself. We see there's a couple of questions in the chat, but before we answer those, does anybody have any questions that they'd like to ask here? I have one. If you don't mind, I'll start off, and then other folks are welcome to raise their hands and I'll unmute you. But how do you see spatial transcriptomics playing into this when we have sort of that all that data available to us as well?
Yeah, I think that's a great question. Did I mute myself or no, I didn't. Okay, good, because I couldn't find the mute button anymore. So in the second version, so I think spatial transcriptomics is crucial to give context to cell annotations. I think it's crucial for a lot of different things. But for the purpose of the Human Lung Cell Atlas, it's really, really important to give context to cellular annotations. And I didn't talk about it at all today here, but we didn't systematically introduce spatial data into the Human Lung Cell Atlas version 1. But what we did do is some of the other data sets, specifically the data set I showed you, which we tested the mapping for, use spatial data to annotate several cell states, which could not be annotated based on single cell data alone. And we find that when we compare, where those cell states that were annotated based on spatial data, when we compare that to the Human Lung Cell Atlas, that information is there. We just don't know it. So without the context, we cannot annotate this well. So spatial data is crucial to give context to the transcriptomic information. And sometimes we just are not as able to understand cellular identity without that context. 61 cell identities should be expandable can be expanded a lot. And in the second version of the Human Lung Cell Atlas, we're also looking to systematically map to space, specifically working together between HCA, LungMAP, and HubMap Lung to kind of annotate this in a broader context and see where we actually need more data to really interpret this better. So.
Great, thanks. Any other questions?
Hi, this is Enid. Malte, that was absolutely spectacular. But I wanted to get a better sense from you as to how you're using the data from some of these really, really important genetics groups like Teichmann's group and others that are really trying to incorporate kind of ethnicity and ancestry variation into the reference genomes. And I don't know how in real time you're planning to do that.
That's nice to see you, Enid. So that's a really good point. So one thing that the HCA as a whole overarching body is trying to do is build diverse reference atlases. We are only as diverse as the data sets we have in there, which unfortunately I think is still over 60% whites and large proportion male, middle-aged. This is the classic enrichment, unfortunately, that we find in the single cell domain. What we're looking to do in the second version is what we've now done already, but it's not too early stage to really talk about it, is to focus on the dataset acquisition, specifically on datasets which would make the reference atlas more diverse in terms of ancestry. Now, that's challenging from two perspectives. One is that single cell datasets from non-white European males, effectively, or European ancestry males, are predominantly tumor studies where we have tumor adjacent tissue as normal. We are going that route now because we think that's the only way to really increase the diversity. Of course, that might mean we have a lot of inflamed cell states, but we can annotate that. The second thing is calling ancestry. So, the typical annotation we get from data contributors is not something we can use. It's very challenging. We cannot get genotype information from everyone either. But there are some really interesting developments in single cell tools which allow you to call at least some level of ancestry information from the single cell reads themselves.
Mhm.
And so we're looking to leverage that to better understand the representativeness across ancestries, and also to model which effects ancestry might have on the transcriptome, which kind of relates then later to, do we find different gene expression patterns in different ancestry populations, which may relate to, drugs working better in particular, or worse in particular populations. And I think that's something we need to start doing. I don't think we're set up perfectly for it, but we're trying to get there.
Thank you.
I hope that answered your question.
I think we probably have time for one more question in this section. And I see Dr. Mark Snyder here has his hand up.
Yeah, can you hear me?
Yep.
That was an outstanding talk. So my question is, there's a growing number of sophisticated algorithms to do cellular deconvolution. However, given this resource here, have you put any thought into giving a more, for lack of a better term, GUI driven process for more precise cellular deconvolution from bulk RNA sequencing data? Because that could be a powerful addition to your tool here.
Yeah. You weren't by any chance a reviewer for HLCA-V1?
No.
Because this is a very helpful comment that we got as well for HLCA-V1. So we added it in revisions. The kind of using the human lung cell atlas to deconvolve bulk data. This is not something we'd initially planned on doing, but I think there's so many applications, we're just not aware of all of them either. So there we started doing this. In terms of the GUI tools for this, it's not something we have at the moment. I think it's a really good shout. I think that the problem a little bit with, so my understanding of the bulk deconvolution field, this is not my main source, my main field of study, but is that, so when we've at least tried to use these tools are very variable in how well they work. There is no tool that is always useful. And there is some level of interpretation and troubleshooting that needs to be done when using one of these tools. That means that makes me a little bit hesitant to say we need to do this, because providing a GUI tool always assumes that you can give people the tools to understand and not give wrong answers without knowing. So I'm comfortable for that with reference mapping, which I think is, even though it's a comparatively younger field, it seems to work very well. And we understand how to evaluate it. I'm not certain I know how to evaluate the bulk deconvolution aspect consistently well. But it's definitely a very useful use case. I just think that the tooling may not be there to be doing this robustly.
That's an absolutely fair response. Absolutely.
So there are a few more questions in the chat. So we're running a little bit behind our schedule. So we're going to move on to the second speaker first. And if we have time at the end, we'll address some of the questions in the chat. So I'm Chao He at the University of Alabama in Birmingham. It's my honor to introduce our second speaker of the day, Dr. Nathan Solomonis from the Cincinnati Children's Hospital, where he is an associate professor in biomedical informatics. Dr. Solomonis has been leading the effort of the HLBI-funded LungMAP initiative, particularly the LungMAP 2 Data Coordination Center. His research aimed to show the hidden splicing single-cell network that underlie normal cell differentiation and disease. And his lab had developed numerous computational approaches to resolve single-cell population and define co-regulatory networks that derive from alternators, transcriptional and splicing program. So without further ado, I'm going to give the floor to Dr. Solomonis. Nathan, I think you are muted.
Definitely have to unmute. Sorry about that. Thank you so much for the introduction. I'm going to hopefully see if I can reshare my slides. I think I am sharing. 1 second. Actually, I'm going to reshare it here. I think it's stopped for a second. Sorry about that. Nope. One moment. I see what went wrong here. Okay. Unmuting is the challenge. Okay, sorry about that. Thank you so much for that kind introduction. I really enjoyed the talk from Malte here, and I'm going to try to jump off some of these topics here and show you some additional resources and really some achievements that the LungMAP consortium has achieved over the last decade now through its 1 and 2 iterations. And now it's about to enter into its 3rd iteration, really focused on understanding the molecular variation that occurs within disease, within specific lung cell populations. And so first, I'd just like to call out really what are the key challenges that we're all facing within lung biology, which are how to really address these fundamental challenges related to regeneration and repair of the lung, especially in disease, understanding the precise role of stem cells in their therapeutic applications, dissecting these complex developmental hierarchies, understanding the more broader and specific mechanisms of disease, and understanding the precise genetics and role of infection in specific cell populations. So these are really major goals and within the community, and addressing these necessitates, at least from the perspective of the NH community, HLBI community, is a team science approach, combining experts with really diverse backgrounds, both technologically, in terms of focus on different regions of the lung and airway, new tools and techniques to construct systems level models, and like Malte showed, is these integrated atlas level views. The LungMAP consortium is 6 centers over the last 10 years. Currently, there are 4 research centers. There is a core human tissue core at the University of Rochester, led by Gloria Pryhuber. This is a way to basically collect rare lung tissue samples throughout lifespan and with health and disease. In a single repository, have really refined metadata and be able to reposit those and send those out to the different LungMAP centers and to the broader lung research community. And a data coordination center, which I co-lead with Bruce Arano here at Cincinnati Children's, along with our co-investigators at the Broad Institute, many of which are involved in the HCA data coordination projects, the UCSC Genome Center, and the Research Triangle International, who are the leads of the DCC in round 1. And importantly, this resource has gotten a lot of adoption through significant citation and use of the resources that we've tried to provide at LungMAP over the last 10 years. And this is a view of some of those resources in terms of their growth, in terms of which unique users that are accessing the website, and some of the unique apps that we've introduced, which have really gotten the most attention, as I'll talk about, these more shiny-based apps, similar to what Malte was talking about. And so really our focus is to create a really robust ecosystem, a web-based ecosystem that enables users from anywhere in the world to be able to take their own lung data and analyze it, to be able to look at our reference data sets, and be able to understand some common, definitive, discrete cell biology within the lung, either at the omics level, at the functional annotation level, looking to see what reagents are available, interrogating complex imaging data in which there's thousands of images for multiple imaging modalities, and being able to even analyze your raw sequencing data from FastQs from scratch, either independently on your own with your own data sets or in combination with the data sets that are provided through LungMAP. And so LungMAP really has this really diverse ecosystem that we've tried to basically encourage the community to use and encourage people to contribute to as well. And it really starts with our ingest broker. And so one of the central focuses of LungMAP is to ingest data sets from our research centers. And so research centers deposit various types of data, whether it be single cell genomics data, bulk epigenomics data, imaging data, metadata, and even really nomenclature and annotations, metadata for different cell types that are emerging through a large number of committees that we have that are across consortia now for specific cell types. But these data and metadata are ingested using a human cell atlas metadata schema to be consistent with diverse dozens of ontologies that we align to. Once aligned to, this compliant HCA compliant metadata model enables reanalysis of this data within these cloud-based workbenches such as Terra, and also allows us to reposit this data not only in dbGaP or Geo where appropriate, but also bespoke dedicated LungMAP data browsers for navigation and analysis, Omero-based, which is an open microscopy environment for interactive analysis of imaging data sets, the central portal, which has a number of different imaging and analysis tools and omics analysis tools, as well as connections to community portals like LGA and TopCell, which are very popular, and this really centralized cell type annotation model, which we've heavily adopted in LungMAP phase 2, which allows us to have these different app ecosystems that are available for community usage for broad omics reanalysis with a focus on single cell omics. Again, please interrupt me if you have questions. I will try to look in the chat, but you also can verbally interrupt me. That is fine. No problem with that. So the really key outputs of LungMAP have been insights, tissues, data sets, protocols, and tools. And many of these are omics-based, and many of these are experimental-based. In terms of insights, we've really focused on what new cell states and regulatory models can be generated from the large wealth of not only LungMAP data, but community data. Again, that we've now centralized on not only the discovery of cell types, novel cell types, but the annotation of cell types. So we have this website. This is lungmap.net. I encourage everybody to go there if you haven't gone there already. You can search based on different types of terms, based on different genes and molecules, and you really are able to get access to a vast array of links from this. But if you were to search, for example, for alveolar duct on the website, one of the links it will take you to is the cell cards resource. And so cell cards, which was originally described in Developmental Cell about 2 years ago, is this growing number of cell types that are highly curated, where there is significant information within the literature to establish what are the functional associations of the perturbations, their existence in mouse and human, markers, imaging data, and genomics links. And so by interrogating these, you can really get access to a wealth of existing knowledge and be able to also interface with the data sets directly. An example of novel cell types that were discovered by our research community is the RAS. One example is the RAS, or respiratory airway secretory cells. And these are secretory cells that were identified within specifically the human distal airway, but not within the murine, that were marked specifically by these secretory specific molecules, and are potentially a way to understand what are different liabilities within diseases that can only be modeled specifically within the human lung. And this is work from Ed Morrissey's group and Maria Basil. I'll come back to this a little later to come back to that one point that I just mentioned. There's also a large number of tissues, which include biopsies, whole lungs. These are available within the linked Brindle repository. So I mentioned the human tissue core. Brindle is the database of the human tissue core. And for each and every sample, once you get a login, so you can see I'm logged in here, you can basically get demographic information on each and every sample that's available to basically order for your laboratory for analysis. There's a diverse number of different types of biological specimens that can be ordered and profiled using different techniques. This includes a diverse number of imaging data that's already collected on these. These include whole lung and even specific sections of specific lungs that are linked to in this Omero imaging viewer that we have, which you can control the intensities of different channels. You can modify these images and customize them and export them to other tools. We also have a heavy focus on omics along with imaging data. And so these are just an overview from, I believe, a little later last year of the different types of samples that are available for human and mouse within this resource. And so you can see that single-cell RNA-seq is the dominant one, but we have an extensive amount of proteomics data and ISH. imaging data for both for mouse. So really there's a significant amount of data that you can correspond between these mouse and human data sets to ask specific research questions without necessarily having to do experiments in some cases. I mentioned the semantic search capabilities within this tool, which include a large number of interactive interfaces. One of these is to explore data sets. You can search for individual data sets here based on different query terms. If you search for a data set, for example, these are the single cell data sets you're presented with, along with the bulk RNA-seq, microRNA proteomics, and others. There's also a number of diverse 2D imaging data sets that are available. And each one of these, for example, single cell data sets, if you click on them and they have these stage 8 descriptors, species, numbers of cells, and diverse link outs, including cloud-based link outs and repository-based link outs. But if you click on one of these, for example, you'll be able to access not only information on the studies, but a number of different tools available. So you can get metadata on the samples, you can get downloads such as those H5AD and other files for analysis. But we also host this diverse number of standalone apps that you can individually query and analyze. And so these include a Shiny Cell browser, which lets you load these large data sets, look at different covariates. You can do similar types of analyses within the cell gene viewer, which Malte showed you for these different data sets. In some cases, we have cell cards linked data sets, as well as in some cases, azimuth viewers, which allow you now supervise the classification of your data sets using H5 files from 10x Genomics, for example, or other protocols or H5AD files to analyze those data sets and align them to our mouse and human references. There's also epigenome viewers, heat map viewers, covariate-specific bar chart viewers, and the raw data itself that you can interface with in Terra. A number of these viewers are also accessible from the cell cards interface where they have been aligned. So not all of our data sets align to these standard reference annotations, these cell card annotations we've aligned to, but for those that are, we have direct links to those on our cell cards page for both mouse and human. The data sets, that single cell atlases that we've constructed in the past and that are currently available on our website were developed through a highly supervised approach. And here, again, we're starting with those cell cards annotations, highly validated, well-defined cell populations with defined markers. And rather than using a large unsupervised discovery like Malte showed, we're actually using a much more conservative set of markers to basically say, what are the well-defined populations based on these marker genes? And so, for example, we have both a human reference atlas with, I think, some 40-some-odd populations, and an equivalent mouse atlas that was spanning development, but now we're working on an adult, as I'll point to in a moment, that you can interrogate at multiple levels. You can visualize your own gene expression data on here. You can look at the underlying specific data sets that these came from, and the specific donors they came from, and evaluate other metadata covariates that are relevant to these individual data sets. We also host a large number of protocols that are both experimental and analytical. So for example, if you go to lungmap.net, there's this resources tab at the top that has protocols linked. If you click that protocols link, you can access the various LungMAP protocols, experimental protocols that have been reposited in protocols.io for community use. This lets you hopefully be able to access different methods for generating spatial data, omics data, or even more discrete specific validations. And we also have similar protocols with our imaging data that is also available at lungmap.net, which includes specifically how those imaging preps were generated. In terms of omics protocols, these include the ability to analyze single-cell omics data and other data types through this TerraBio.cloud-based environment. It's fairly cheap to basically analyze data sets. You can analyze them on the pennies, typically. And you can use our own repository data sets. And if you have dbGaP access to our controlled access data sets, you'll be able to use these and reanalyze these data sets in a fairly streamlined way through the use of these tools. There's example tutorials. This is one of them for reanalyzing single cell data sets that have been reposited in LungMap. But there are other protocols I'll point to in a minute. There's also this omics data viewer that I mentioned before, this omics data browser. And this omics data browser is really the way you access the raw data and process data. So this is really a scaled environment for if you go and you collect into these links, you'll get detailed metadata on the individual samples. You can download that metadata. You'll be able to access the individual FASTQ files or download them or reanalyze in the cloud along with any of the process files that exist for these data sets. I mentioned the Omera imaging browser. This really has a diverse number of developmental time points and technologies, including immunofluorescence confocal, histological stains, and a number of emerging high throughput visualization technologies, all which have advanced metadata on them that lets you go back and see exactly where they're from and be able to link back to the lungmap.net website to see what the detailed metadata on those are. And there's a large number of other tools, including protocols for organoid production. I think organoid data sets are now available online at lungmap.net, omics and informatics. We've also developed a number of tools within LungMap for analysis, specifically that are standalone tools. And these include a tool I'm going to mention in a moment called SC Triangulate, Cell Drift, which lets you do temporal perturbation analyses, Udon, which lets you understand what are disease-specific novel subtypes when you look at a single large collective single-cell study or different single-cell studies combined, as well as tools for spatial transcriptomics and flow cytometry analysis. And these are on top of a number of tools for data visualization, such as that Omero portal I mentioned, and the TopCell browser, which I haven't mentioned, which lets you access dozens of lung data sets to explore different lung-specific gene signatures and cell type-specific signatures. And importantly, we encourage people to contribute to this resource, the LungMap, through the LungMap ingestion broker. So you just need to contact us and say, we'd like to basically have this data set in LungMap, and we'd like to submit it to you. So please reach out to us, and we can work with you to onboard you to that process. Under resources is this tutorials tab, which I encourage you to visit. There's a number of both video tutorials and step-by-step tutorials. Again, community contributions are welcome, both for in terms of these tutorials, in terms of use cases, and in terms of recommendations that you would like us to have in terms of functionality on these websites. So now I'm going to move towards really what our goals are for the next stage of LungMap, which is developing these multi-omic disease catalogs. And so within LungMap Phase 3, our mandate is to develop basically large cell atlases that comprise both within and between disease comparisons to understand where there is commonly shared basically cell states or disease programs that span defined diseases or other conditions of interest. This is using both single cell and spatial omics approaches, although the field is evolving, and if there's new approaches that we can adapt, we will. And importantly, new approaches to understand the gene regulatory landscape. So what are the regulatory transcription factors where the genomic open accessible regions that transcription factors are binding to, and can we basically infer these gene regulatory relationships from large data sets such as single-cell RNA-seq, single-cell ATAC-seq, multi-omic, and single-cell chip-seq technologies that are now emerging. This doesn't go without challenges. As all of you know, these are very difficult questions, and Malte presented some very nice tools and approaches to start addressing these questions. But you have you're limited both in terms of the study cohorts you have and their designs, which is a major challenge. These include batch effects, ethnicity, treatment protocols, technologies. Importantly, data exists behind different firewalls, and it's very hard for people to get access to those. dbGaP, ArrayExpress, you have different firewalls that limit your access to these data sets. There's a lack of harmonization. You often are faced with downloading preprocessed data sets which have different gene models. They're generated at different time with different initial tools and QC and data cleaning like ambient RNA correction. And there's a lot of different automated integration approaches that have variable performance that create a number of challenges for analyzing these data. We can't solve all these problems, and so we're really pointing to the community to work with them to find the best solutions. A number of those solutions were mentioned Malte here. But one approach that we've been working with is a tool we recently developed called SC Triangulate. And the goal of this tool is to take data sets and to not only basically identify the most robust cell populations, and by robust, I mean durable, stable, reclassifiable with unique marker genes, but be able to do this across different modalities. So if we have multimodal data with RNA, attack, cell surface, protein-based markers, we would like to be able to consider that multimodal data. We'd like to consider different input algorithms in terms of the clustering, but we'd also importantly like to consider different prior annotations that have been generated. So if we have HLCA annotations, we have LungMAP annotations, we'd like to be able to integrate these different annotations, and somebody may have found a rare cell type somewhere and annotated it correctly, but how can we leverage all these diverse annotations in a coherent way? And so we developed a game theory focused approach that is basically looks at different stability metrics to identify the most stable populations over potentially dozens of different input possible solutions. And this program then identifies the most discrete set of durable populations from the integration of potentially dozens of these data sets. And so this was a proof of concept that we initially published in this paper. Here we're integrating results from 4 different proposed atlases. And here we're actually mapping those annotations onto a single reference atlas. This is a single nuclear RNA-seq data set as just to initially evaluate with its initial annotations. We project these different data sets on there. Some annotations win out over others for different cell populations as being the most stable, and we have associated confidence metrics associated with that. And from this, we have combined integrated atlases where these annotations are not actually, we're not manually assigning these annotations. These are labels that are coming from these different prior cell atlases where they're providing the most stable results. And by doing this, we basically can identify, in this case, for example, these different dendritic cell populations that derive from 2 different data sets, but were found in combination to be the most stable. Similar for these alveolar macrophage populations, one that was called MAC alveolar, the other was called alveolar MAC. But these clearly have different transcriptomic profiles within the same data set. And importantly, we also can benchmark these to solutions such as HLCA and to our own LungMAP cellref to see, can these be validated with kind of integrated, independent integrations on independent data sets and annotations. And so we've been now using this type of approach and taking this to the mouse and in this case, we take 22 geo studies where there's mouse single-cell RNA-seq data sets. We don't have those firewalls I was talking about before, which are challenging to get across, but we download the raw data. We have over 250,000 cells. We take all these different annotations from different sources, and specifically we take our own cellref, human cell ref annotations and our mouse cell ref annotations from our azimuth instance and project those onto these combined data sets. This gives us 85 combined clusters. We also perform unsupervised clustering with tools such as ICGS2. And we get a similar number of annotations, and then we can integrate all these annotations and say who wins out, and we get this final result. And so you get a large number of populations, which you should be skeptical about if you're looking at that these are real. But importantly, by integrating these diverse studies, we're able to capture cell populations we normally would never really isolate. So for example, we have Schwann cells, and lo and behold, this comes from a study which they profiled Schwann cells from the lung specifically. So there are a lot of focus captures in here for very specific cell compartments, including secretory cells and tools and cell types from the airway, which are rarely captured in most murine lung studies. And these annotations really are most stable that come from different sources. So for example, the human cell ref is finding, is most stable for these AT2 populations, where the mouse cell ref was actually identifying these discrete DC and IM subtypes or cap cell subtypes and others. But the majority of populations were identified by this unsupervised clustering. And one highlight here is that RAS cell population I mentioned before, which has not shown to not exist within those distal alveolar respiratory bronchioles. And here what we're identifying is, as we see that same marker that was defining RAS cells in human, but we see it in mouse. But importantly, these cells, when we ask where they're coming from, are specifically coming from these really focused enrichment analyses of a specific airway population that you normally would not identify unless you really did these focused captures here. And this is these are larynx cells, and in addition to that, you also see that they're uniquely representing for specific populations. So by basically enriching for these populations, you can now combine all these data sets together with these focus captures and potentially identify either known or novel or recurrent biology across different studies. And so our challenges going forward are really to, be able to not only generate these large cell atlases and account for disease heterogeneity in cell populations, but also understand broad transcriptomic impacts and disease gene programs that are now affected with these reference atlases that we have. And so one approach that we recently developed to do this was called UDON, which is unsupervised discovery of novel gene programs. And it basically, this approach, you supervise reference cell annotations, you then subdivide these by the original donor annotation, you collapse these into what we call pseudobulks, or the average gene expression per donor and per cell population. We have these for both healthy and disease. We're comparing the disease. You can compare disease to specific matched healthies within that matched cohort or across cohorts as a combined pseudobulk. And then once with these in hand, we then can compute differential gene expression for each individual cell population pseudobulk and each individual patient. And by doing so, now we can perform unsupervised clustering on the differentials themselves to see if there are heterogeneous new subtypes of disease that we can identify across different single cell populations and even across heterogeneous diseases. We initially applied this within the context of a disease called SJIA, or systemic juvenile arthritis, which has a significant lung disease component here, as you can see in this UMAP, where there are each one of these purple dots represents a different pseudobulk. So one patient in one cell population. And we can see these different pseudobulks basically run together for different types of diseases. For example, active disease versus this macrophage activation syndrome versus inactive disease. And we get these different subpopulations that are clustered with these different associated functional associations that are derived through gene set enrichment. And what we can learn from this is we can understand what are the discrete gene programs that are shared among different disease subtypes and effectively identify novel disease subtypes that span different types of diseases or related diseases in this case. And importantly, by itself, you can't validate this. How do you go validate? You do validate this in an independent cohort, which we've done. But you can also take these large bulk RNA-seq data sets, or in this case, bulk microarray data sets, and you can say, do we see independent evidence of these gene programs, these UDON subtypes, within a larger group of patients? So I believe this is over 100 different kids with SJIA, which you can evaluate here with microarrays versus bulk RNA-seq, I believe. And you can actually look at these disease subtypes and see if they mirror what we see in terms of subclinical subtype associations. And so this lets us even go a little bit deeper. So we can now start looking at specific UDON clusters. We can say what genes are basically driving these different gene programs within these UDON subtypes. And these, in some cases, for example, this was highlighting complement activation as a potential regulator of a unique subtype of SJI disease. And we can validate that now with serological measurements we have that are matching on the same or independent patients. And finally, once we have these UDON subtypes, we can even supervise them across related diseases. In this case, we related them to SLE. And we can say, can we pull out similar SLE subtypes within either those diseases or even within potentially within healthy individuals from large single cell compendiums, such as this 1K PBMC cohort, to say, are there rare patients that have these potentially early stage disease programs? So we're using a lot of these different tools going forward, but we really need significant contribution of the community and our LungMAP team. So this is the current LungMAP DCC leadership team, we're entering into phase 3, which includes a lot of computational biologists, coordinators, and technology advocates that are really pushing these resources to different cloud-based ecosystems. And with that, I'm happy to answer any questions.
Thank you for the great talk. So if any of the audience have any question, please raise your hand and unmute yourself. And or if you prefer, you can put in the chat box here.
So I'm just entering the resource lungmap.net over here on our webpage, and there are a number of video tutorials that I'll just also hop into the chat there, which I recommend for using these different tools.
Yes, we do have a question in the chat from Sadia Saki, who's asking about the red cell. So the red cell identified in human data set, was this identified in neonates or was that in adults?
Yeah, that's a great question. I believe I'm trying to actually, I have to look back at that. That's a good question. You caught me a little bit off guard because I'm not as familiar with that paper as I should be right now. But looking back, yeah, I can't recall if it was specifically from neonates or adults, so I don't want to misspeak there. It was a population that was specifically impacted in COPD. And it's thought to be specifically, I believe, lost in COPD. So it should be in adults, but I don't remember where it is most enriched. Yeah, we definitely, we're using data from a highly enriched protocol to identify those cells in the mouse. But I will put the link to that study in the chat.
Is there any other questions from the audience, or Dr. Liu can stay online? So if any questions about the first presentation.
Yeah.
Yes.
I have a question. So I'm interested in the rat cells you show in the map. So how different, like what's the main difference between this rat cell with the club cell? Because I see it's a very high press of the SGB1A1. So are these cells also express the SGB3A1 and the SGB3A2?
Yes, they were coincident for both, and I believe they were more specific for SCGB1A3 than they were, or is it 3A2? Sorry, 3A2,
Yes,
is that right? Sorry,
yes.
3A2 than the SCGB1A1. And so I should note that when we identify the cells, the reason we have confidence in assigning the name RAS to them is we're actually using the human azimuth on the mouse data set. And so we're basically projecting the labels, and these were projected to be RAS cells. These were predicted to be RAS-like based on the gene set from the RAS. But these were predicted to be RAS cells from the azimuth projected labels.
Yep, thank you.
Yep.
I just want to say, wonderful, Nathan, wonderful, Malti. I mean, these are such critical talks at this transition point in lung biology, where these tools are going to really kind of create a foundation for work that we'll have to do for the next 20, 25 years. So I thank you both. This has been incredible.
Thank you.
Thanks.
Yeah, I think we're at the top of hours. So on behalf of the RCMB web committee, I just want to echo what Dr. Neptune said. Really great talk from both Marte and Nathan. And thanks for your time. Particularly Marte, is already almost 10 o'clock in Germany, 10 p.m. in Germany. So just kind of want to remind everyone the video recording of this talk will be available on the ATS website. So if you or your lab colleague want to kind of review the talk, scan the QR code and try it on yourself, you're free to review the video recording, do it on your own. But thank you, everyone. Thanks for our speakers. Everyone have a good day.